Let's have a quick recap about the terminologies that we find on phylogenetic tree. So we've got this one's a general picture where you see the beginning of the tree is the root, the top is the end bits here. They're known as the terminal taxon. So each of these are taxons or taxa. Two taxa, which are on the same node, are known as sister taxa. And if you have more taxa on a node, they're known as polytomy. So there are many taxa there. And this is a node, this is a branch, and you can have trees that are rooted and unrooted. Don't forget that the trees can also come in different formats. You can have them as polar or circle format or radial format. Notice the red dot, and that's where the root of the tree would be. Quick look at what data is usually shown in a tree. The trees we'd looked at before did not have these distance values. So let's have a quick look at what those values actually mean. The horizontal lines are branches and represent evolutionary lineages over time. And this is interesting because the other trees we're looking at, they just showed you the relatedness of all the organisms on that tree. When you have these distance measurements, it gives you a better idea of what's really happened at a mutational level. The longer the branch in the horizontal dimension, the larger the amount of change. The bar at the bottom of the figure, this one, gives us, well, a scale for this. In case that, like in this situation, you've got 0.07. So this tells us that the amount of genetic change is 0.07. That also relates to a specific nucleotide site. So that is the unit branch length is usually nucleotide substitutions per site. That is the number of changes or substitutions divided by the length of the sequence, although it can also be given as a percentage change, in which case you're saying that there's a certain number of changes per every 100 nucleotide sites. And that should give you a clue as to how much change has really been happening. Make a phylogenetic tree using UGENE. So we go to UGENE, we open the file, and let's look at our BLAST results. And when you open them, remember we downloaded, was it 4 or 5 separate files from our BLAST search? So you can either open them as you just merge them into a single sequence, or what we want, join the sequences into an alignment, and then you open them in the multiple alignment viewer. So if we choose that, this is what you're going to get. So these are our sequences. They've all been aligned. And if you see what an alignment is it just tries to join nearest neighbors. It looks to see the similarities in the sequence and tries to put them all together in a way that matches the similarity best. So you can see here, all the base pairs that match are all in the same place. So like this one here, you can see there's T's all the way through. So the first, if you just look at this, you can see by looking at the colors, you can see the blocks where the colors are all matching all the way through. Like here, that means that there's an alignment at this position through all of the sequences. But if you look a bit further, like let's say where there's 3 alignments here. Here there's all 4 sequences are the same at that position. So by looking at these similarities, you can see which parts of the sequence are most similar, which ones are most different. Just from a quick glance here, we can see that this one, and this one are more closely related to each other than this one. This one seems to be quite different. You see how the colors don't match what's going on in the other 3, whereas the other 3 seem to match each other. So you could do this exercise with your eyes and try to see what's similar, but it's difficult. So that's why we use this software. But just from this quick glance, you can see that this one is the most dissimilar, whereas these ones are most similar to each other. But even as they're similar to each other, there were a few differences that we spotted along the way. So let's see how this looks in a tree. All you have to do is go up here. There's a tree. So you click the tree, and when you do, it asks you what kind of method you want to use to build your tree. This Philip method is the easiest to use. It just uses neighbor joining, which is an algorithm. There are all these different types. It's a distance matrix. For you, when you get into bioinformatics, that's when you would need all these other different types of algorithms. For here, we just use Philip, and you tell it to go. And when you do, it will generate a tree for you now, so you can visually see and also look at the distances. So here's the tree. Let's see if we can get it a bit bigger. I'll try to move this over. Here's the tree. So remember the tree from the other one? This one has got the distances as well. At least you can see the distances, the distance between the two sequences. Here there's 0, meaning these two, there's no distance between them. They're basically very much the same as each other, whereas you have these other ones with the distances there. You can also choose whether you want it to be a rooted tree or not. So you can reroute the tree using different parameters. As you start doing trees, you'll see what's important about that. But one of the things that you also need is to choose the right outgroup. So if we go back to our exercise, oh, by the way, you can also generate a picture of that by just getting the camera there, and you can save it as an SVG or as a PNG. You can copy it to your clipboard and then put it in your assignment, wherever it is you need it. So if we go to the exercise, we were looking over here. This is this consensus sequence, I think just before we zoom on. Sorry to take you back. The consensus sequence was what we generated. Remember when we're saying the consensus between the different sequences that we looked at. So when we looked at the hits that we got from the blast, we got the two which were close to each other. So this would be your consensus sequence right here. Likewise, if you look on UGENE, and we had this. So this becomes more important when you have very dissimilar sequences. So usually the consensus would be right at the bottom. Need to get this a bit more. There. So there's can you see here it says consensus? So this is where it's telling you that this is the consensus between all the sequences you've given me. In general, you have this sequence here, and these are the places where there's a lot of consensus. They're the same basically. And then there are places where it's very different, where there's no consensus at all. Or there are places where the sequence missing, there's a gap. So you'll see that when you do blast searches, there might be two places where the sequences are highly similar, but in one species, there's maybe a gap of 10 bases between the place where the two sequences match again. So all that will be shown in the consensus sequence. So this is either shown up here or sometimes down at the bottom, but it will say this is the consensus. So that's what we're talking about when we say consensus sequence.